Hello everyone, welcome to another topic from DNA Bioscience. Today we are going to discuss about the complement systems. So let's discuss. Complements are generally the group of plasma proteins. Mainly 20 plasma proteins are there which helps in the innate immunity. If you don't know what is an innate immunity, please go to my innate immunity video. Then you can understand the complement system in a better way. So the name is complement as it implies complement, or you can say it enhance our immunity. The organ where the complement system synthesized are the liver. So liver is the organ where the complement system synthesized and it then distribute to all other the tissues and the body fluids. As I said, 20 plasma proteins are present, but out of 20, only 11 proteins are mostly involved. The B, the C1 to C9, that is C1, C2, C3, C4, C5, C6, C7, C8, C9, and the T protein. So let's discuss in detail. The first one is the classical pathway. The name is classical pathway as it is introduced later. So it is activated mostly by the antigen-antibody reaction. Question may come that which complement system involved the antigen-antibody reaction. So the answer is classical pathway. So after the antigen and antibody interaction, they activate the proteins, that is C1, C2, C3 and C4. And as I said, it is antibody mediated, so it wait for the immunoglobulin M or immunoglobulin G. So first it activates the C1 protein complex, and the C1 protein complex, it is a hexamer. It consists of C1q and a serine protease, that is C1r and C1s. So in the first step, the C1q binds to the Fc region of the antibody and it cleaves the C4 protein. The C4 protein splits into C4b and C4a. The C4b protein then binds to C2 and form a convertase that is called C4b2a C3 convertase. Then in the similar way, the C3 convertase, it splits the C3 protein to C3a and C3b. The C3b then binds to the C3 convertase and forms the C5 convertase. In this way, it forms the C6 convertase, C7 convertase, C8 convertase, C9 convertase, and the final product is C5b6, 7, 8, 9 and form the membrane attack complex, which leads to the cell lysis. So this is all about the classical pathway. Let's move to the lectin pathway. So the lectin pathway, it involves the mannose binding lectin. So the question may come, which pathway consists of the mannose sugar? So the answer is lectin pathway. So the pathway consists of mannose binding lectin, C2, C3 and C4 proteins. With that, some calcium dependent lectin family proteins. So the MASP1 and MASP2 bind to MBL, that is the lectin associated serine protein 1 and serine protease 2 bind to the mannose binding lectin, which activates the C4 and C2 protein complex. So as we have discussed in the classical pathway, in the same manner, the C4 after activation, it splits into C4b and C4a. It splits to C2b and C2a and it forms C4b2a, that is called C3 convertase, and as we have discussed in the classical pathway, it forms the C3 convertase, C5 convertase and last the membrane attack complex. So this is all about the lectin pathway. Let's move to the alternate pathway. The name is alternate pathway as it is totally different. It involves the factor B and factor D, which is a kind of complement enzyme. So let us discuss. So generally in this complement system, the hydrolysis of C3 complex occurs in the blood plasma. So the C3, it divides into C3A and C3B. The C3B, then in the presence of some calcium and B factor, it forms the C3BB, and then by the help of factor D, that is a complement enzyme, it forms the C3 convertase, and in the same way it forms the C5 convertase. Up to C9 convertase it will form, and then it forms the membrane attack complex and results in the cell lysis. So this is all about an overview of complement system. You need to understand first, in the classical pathway it is an antigen antibody interaction and where the C1 complex is required, and in the lectin pathway the main thing is the Mendon binding lectin and the MASP, and in the alternate pathway the main thing is the factor B and the factor D. So let's discuss the effects that caused by this complement systems. So the first effect is it results in opsonization. Generally due to the C3b which is present in the alternate pathway, it activates the antigen presenting cell which results in phagocytosis and opsonization. The next thing is the cell lysis, that is the membrane attack complex. In all this system, at the last, membrane attack complex is formed. So as the name indicates, the membrane attack complex, it attack to the wall of the cell, so it leads to the cell lysis. Then the next point is the chemotaxis that occur due to the C5, and the fourth point is the activation of the mast cells. We have already discussed about the mast cells in type 1 hypersensitivity reaction. So the C3a, C4a and C5a, they are mainly helpful to activate the mast cells. So this is an overview of the complement system. I hope it will help you, and please subscribe to my channel to get more such videos. Thank you.